cells, which are in your skin. You have muscle cells, which are in your muscular system, and they're composed of not only skeletal muscle, but smooth muscle cells. You also have fat cells. You also have white blood cells or macrophages. You also have neurons or nerve cells, and also cells for reproduction. So, what are the common structures for every cell? Well, human cells have three basic parts: a plasma membrane, which is the outer boundary, which we're going to talk about today. We're going to focus today's lectures, there are two of them, part A and part B, on the plasma membrane. Then on Wednesday, we will talk about the cytoplasm and the organelles that this fluid contains, and we'll also talk about the nucleus, where the DNA is, where the control center of your cell is. So know the plasma membrane, cytoplasm, and the nucleus. You have a lab associated with this course, and I'm sure in the lab you have seen this diagram, which we'll talk about. These are the organelles that are present inside the cell. We'll talk about them in greater detail on Wednesday. So you have to separate substances that are found outside the cells and substances that are found inside the cells. Substances that are found outside the cells are called extracellular, outside of the cell. And these basically are body fluids. Interstitial fluid is the fluid between cells. Blood plasma is the fluid that the blood cells swim in. Cerebrospinal fluid is the fluid that surrounds the nervous system. Cellular secretions include mucus and saliva, and basically this forms a matrix. This extracellular matrix is the glue that holds all cells together. Now, let's talk about the plasma membrane. The plasma membrane is a barrier separating the outside of the cell with the inside of the cell. The ICF, or the intracellular fluid, is inside the cell, and the ECF, the extracellular fluid, is outside of the cell. The plasma membrane is also known as the cell membrane, and it plays the most important role in things that come in and out of the cell. It is the barrier. It is composed basically of lipids. When you say the word lipid, you mean fat. And on this bilayer, there are membrane proteins that float through the fluid membrane, constantly changing its patterns. Also on this membrane are sugars, and they're on the surface, and they form what's called a glycocalyx. Glyco means sugar, calyx means skeleton, so there is a sugar skeleton formed by these sugars around the cell membrane. The structures hold these cells together through cell junctions, and there are three types of cell junctions, and we'll talk about those junctions later. The lipids, the fats, it is a bilayer. It is composed of 75% phospholipids, which consist of two parts. The head, which is the phosphate, is charged. And when you say the word charged in medicine, it means they are polar. So they are water loving. The word hydrophilic means water loving. Hydro means water, Philic means loving. The city of Philadelphia is the city of brotherly love. That's how you can remember hydrophilic, water loving. The tails are made of fatty acids. They are nonpolar. They have no charge. They are hydrophobic. Hydro meaning water, phobic meaning hate. Hydrophobic, water hating. The remainder of the bilayer is composed of 5% glycolipids, lipids with sugars added to them. And the rest is 20% cholesterol. And the cholesterol increases membrane stability. You may have heard in passing of good and bad cholesterol. Well, this is good cholesterol. And here it is. There is a polar head, which is hydrophilic, and a nonpolar tail, which is hydrophobic, loving water, hating water. On the membrane, there are membrane proteins. These membrane proteins communicate with other cells and the environment. They have membrane function, and there are two types: integral proteins and peripheral proteins. Integral proteins, they are firmly inserted into the membrane. They function as transport proteins. They bring things in and out of the cell. They create channels. They create carriers. That is their main function, to transport proteins in and out of the cell. The peripheral proteins, they are attached to the integral proteins. They basically function as enzymes in chemical reactions. So the integral proteins function as transport carriers, and the peripheral proteins function as enzymes. Transport. Basically, those are the integral proteins, okay? That's important. That's important. And the other ones are enzymes. They function as enzymes. So know the difference between integral and peripheral proteins. You dig into this way too much. You don't really have to know all the intricacies. Just know the differences between integral and peripheral proteins. The glycocalyx, the skeleton of the membrane, it consists of sugars sticking out of the cell surface. They are attached to fat or lipids. Therefore, they're called glycolipids. If they're attached to proteins, they're called glycoproteins. Each cell has a different pattern of this coating, otherwise known as sugar coating. It allows the immune system in your body to recognize self or non-self. If there's a foreign protein from the outside trying to get into the cell, these sugars will identify it and block it. Not only will they block it, they will alert white cells to come down and destroy it. So they identify self and non-self, the sugars, the glycocalyx. And as you can see, it's an important thing. If your immune system is acting correctly, it can dispel cancer. If there's a virus that causes cancer in your body, the glycocalyx will identify that virus and kill it. before it gets into the cell to produce cancer. So that's very, very important to understand. Cell junctions. There are three of them. There are three ways that cells can be bound to each other. They can be bound to each other by tight junctions, desmosomes, or gap junctions. Okay? The majority of cells are bound to each other. The ones that are not bound to each other are free. And those free floating cells are blood cells and sperm cells. The other cells are bound together to form organs and tissues. Tight junctions. Tight junctions are where the integral proteins fuse to form an impermeable junction that surrounds the entire cell to prevent liquids and most molecules from moving in between cells. There's a question here. Where might these be useful in the body? Well, your skin. The epidermal cells of your skin don't allow water to get into your skin. It allows oxygen and carbon dioxide to go in and out, but it doesn't allow water to get in. It does allow water to come out because when you sweat, those tight junctions open up and let the sweat come out. So tight junctions are basically in the epidermis, your skin. Okay? Desmosomes. Desmosomes are basically zipper-like junctions that look like a zipper. They have interlocking teeth. Basically, this junction is in between cells, but allows the cells to give a little bit, to allow stretching. Desmosomes are when you have an organ or a cell that's under a lot of tension, like in the gastrointestinal system. When you eat too much, what does your stomach do? It enlarges. How about when you have to go to the bathroom and you have to hold it so your bladder fills up more and more? That's where desmosomes are. Tension, tension, zipper-like junctions. And the last one is gap junctions. Gap junctions are in proteins that form tunnels to allow molecules to pass from cell to cell. Basically, it's the allowance of electrical signals to be passed. And when you talk about electrical signals, you always talk about the heart, because the heart is based on electricity because it has its own pacemaker, and also other smooth muscle cells which are in your arteries and veins. So here you have the third junction, gap junction. These junctions constantly allow substances to move in and out of the cell through the plasma membrane. The plasma membrane is selectively permeable, allowing only certain molecules to cross and stopping molecules that you don't want inside your cell. There are two ways that these substances cross the plasma membrane: either passive transport or active transport. The word passive means no energy is required. Active transport means that energy is required. And whenever we say the word energy in biology, it's ATP, adenosine triphosphate. That is how you use energy, by hydrolyzing ATP. Passive transport, no energy, and no ATP is required. Let's talk about passive transport. Passive membrane transport requires no energy, and there are three basic mechanisms of passive transport: simple diffusion, facilitated diffusion, and osmosis. All of these involve diffusion, a natural movement of molecules from areas of higher concentration to areas of lower concentration to create equilibrium. And what we say is they're moving down a concentration gradient from high to low to maintain an equilibrium. All these molecules have a high speed movement due to their own kinetic energy. You don't need ATP with passive transport. Here's a typical example of diffusion. If you drop a concentrated pellet of dye into water, eventually it will dissolve. What it's doing is equilibrating with the water. And eventually the dye will be evenly distributed in the water. That is simple diffusion. Simple diffusion. Diffusion is influenced by three factors. Number one is concentration. The greater the differences of concentration between two areas, the faster the diffusion occurs. Molecule size. Obviously, smaller molecules diffuse across a membrane faster than larger molecules. And the third is temperature. Higher temperatures increase the kinetic energy, which results in faster diffusion. Equilibrium is reached when there is no longer a movement in either direction, inside or outside of the cell. That's known as homeostasis, equilibrium. Your plasma membrane is differential permeable. Remember, its nonpolar hydrophobic lipid core will stop diffusion and create concentration gradients, acting as selectively permeable barriers. So not everything will get into the cell and not everything will get out of the cell because it is selectively permeable. Molecules that are able to passively diffuse through a membrane is known as simple diffusion. They don't need channels. They don't need energy. Simple diffusion. Oxygen and carbon dioxide are gases that will go in and out of your lung without energy. If there is a larger molecule or a polar molecule that has trouble crossing the membrane, it can be assisted by carrier molecules produced by the membrane. And that means that it is facilitated diffusion. These carrier molecules attach to that larger nonpolar, soluble, or polar molecule and pull it in, or take it out. The special name for this movement of solvent, usually water, is osmosis. So when you have the movement of water in and out of the cell, it's known as osmosis. Not molecules, but water. Water is the solvent the universal solute of all cells. If your plasma membrane is severely damaged, substances will just leave the cell and substances will get into the cell. This happens with burn patients. When your epidermis is severely burned, you lose fluid, you lose protein, you lose sodium, you lose potassium, you lose chloride, you become dehydrated, and you can easily die unless the physicians and nurses give you back that fluid and those enzymes and those electrolytes. Simple diffusion. Simple diffusion is gases, oxygen, carbon dioxide, also steroid hormones and fatty acids. But the main thing with simple diffusion is oxygen and carbon dioxide. That is very, very important to understand. Simple diffusion, no energy. It is passive transport. Facilitated diffusion, you need help. These molecules are too big. Glucose, amino acids, some ions are too big to passively be transported. So they need to bind to proteins, or they need to find a channel through which to go through. So you can either have carrier-mediated facilitated diffusion, which means they're bound to protein carriers, or you can have a channel-mediated facilitated diffusion, which means they move through water-filled channels to get in and out of the cell. Carrier-mediated is by the proteins that are the integral proteins on the transmembrane. Remember, we talked about integral proteins. Well, here they are. Integral proteins are the carriers in carrier-mediated facilitated diffusion. They bind molecules, they envelop them, they change shape, and that results in a molecule being moved across the membrane in the process. Carrier-mediated facilitated diffusion. Okay? They can have multiple problems, and they come through channels. They can come through channels. So if they can't come through a carrier, they come through channels. And there are two types of channels that they can come through. Leakage channels, which are always open, and gated channels, which are controlled by chemical or electrical signals. Basically, these are all water channels. And those water channels are known as aquaporins, for channel-mediated facilitated diffusion, known as osmosis. Whenever we transport water down a concentration gradient, it is known as osmosis. Okay? And this is an example, and you can look at that at your leisure. But osmosis is the selectively bringing in water, not molecules, in and out of the cell. And there it is again, twice already. They go through specific water channels called aqua, which means water, and porins, which means holes, water holes. They go through them. There they are. Osmosis. Osmolarity, you don't really have to know. It is very complicated, and you don't really have to know it. It will just blow your mind. Don't worry about it. Know the word osmosis. Know what it means. Don't worry about osmolarity. All right? This kind of shows you the equilibration of molecules and water. Eventually, if this is the membrane and there's a lot more solute, which are molecules on this side, than there is on this side, eventually it will equilibrate. It will equilibrate. The higher concentration gradient of solute will go into the cell through the freely permeable membrane, and you now have equilibration, known as homeostasis, equilibrium, equilibrium. Osmosis is, we're talking about water. We're talking about water. Water will have net movement across the membrane until they are equal. That equal term is called osmolarity. You don't have to worry about that, but you need to know it goes from a higher concentration to a lower concentration, and then it will equilibrate. It will equilibrate. All right? There are also hydrostatic pressures and osmotic pressures. You don't have to know that. That's not important in this. It's not important. Don't worry about it. Okay? But what you do know and should know is that if water leaves the cell will shrink. The cell will shrink. When it shrinks, it has a disruption in cell function. If there's too much water that gets into the cell, it will swell. What happens if you blow up a balloon with water? What happens eventually to that balloon? It will burst. So too much water in a cell will make it burst, and too little water in a cell will make it shrink. Okay? They talk about tonicity. You don't have to know about tonicity, but you certainly have to know about shrinking. Shrinking is referred to as crenation. Crenation. Shrinking. Water flows out of the cell shrinks. It crenates. When water flows into the cell bursts. That's called lysing. And there's an example here, I think, of a red blood cell in a minute. Here it is. Here it is. This is a normal red blood cell, a nice biconcave disc. Okay? When you have water leaving the cell, it crenates. It crenates. And the reason it crenates is because water has left the cell, because outside of the cell needs water. So water will leave the cell and it crenates. If outside of the cell contains too much water, then the water will get into the cell, and if too much water gets into the cell, this red blood cell will burst. And that's called lysis or lysing. So this is crenation, shrinkage. This is lysing or bursting. Too little water, too much water. Any questions on that?
Nope.
Okay, here we go. Second part. Second part is part B. We went through passive transport, and now we're going to talk about active transport. There are two mechanisms or processes for active transport. One is pure active transport, and the other is Vesicular transport. The word vesicular means bag. That means they're transported in bags. When you say the word active transport, you need ATP. Why do we need ATP to move things in and out of the cell? Well, the solute, which is a molecule or a protein, is too large. The solute or molecule is not lipid soluble, not fat soluble. The solute is not able to move down a concentration gradient. Remember, the concentration gradient is from a higher level to a lower level. This is passive transport. If it's not able to move down the gradient, you need ATP. So you need ATP for all three of these. Active transport requires protein carriers. These protein carriers are known as pumps, solute pumps. They bind specifically and reversibly with the substance being moved. Some carriers transport more than one substance. Antiporters. Antiporters transport one substance into the cell while transporting a different substance out of the cell. Symporters. Symporters transport two different substances in the same direction. In active transport, you're moving against a concentration gradient. You're now moving from low to high. So you need energy. And that energy is always ATP. There are two types of active transport. The only one I want you to remember is primary. I don't want you to remember secondary. I'm not even going to talk about it because it's irrelevant. What's irrelevant is primary active transport. It requires energy directly from the hydrolysis of ATP. Primary active transport is energy from the hydrolysis of ATP. It will change the shape of the transport protein. A typical example of primary active transport is the sodium-potassium pump. The sodium-potassium pump is well known. It basically is an enzyme, and that enzyme is sodium-potassium ATPase. It pumps sodium out of the cell and potassium into the cell. It's located on all plasma membranes, but it's especially active in excitable cells. And those excitable cells are nerves and muscles. It is based on leakage channels. It will leak sodium out of this into the cell and leaking potassium out of the cell. The potassium pump works as an antiporter. It will pump sodium into the cell, out of the cell, and potassium back into the cell. So if you lose potassium, you need the sodium pump to put the potassium back into the cell. If you put the potassium back into the cell, you need to take the sodium out. So the sodium-potassium pump is an antiporter. One goes in, one comes out. Antiporter. It is the basic function of muscle cells and nerve cells. The sodium-potassium pump. primary active transport, requires ATP. You're going from a low concentration gradient to a higher one. If you do that, you need energy to do it. So you need ATP. Okay? 3 sodiums bind to the pump protein. The sodium binding promotes the hydrolysis of ATP. Energy is released, and it adds a phosphate to the pump. The addition of phosphate causes the pump to change shape, expelling the sodium out of the cell. The 2 extracellular potassiums bind to the pump. This potassium binding triggers the release of the phosphate. When you lose the phosphate, the pump resumes its original conformation. The pump protein then binds with ATP and releases the potassium to the inside. The sodium sites are ready to bind again, and the cycle continues. It brings potassium into the pump and throws the sodium out of the pump. Got it? Secondary active transport, you don't need to know. You do not need to know it. I'm not even going to talk about it. I'm going to skip right through it and go to vesicular transport. Vesicular transport involves the transport of very large molecules called macro, which means big molecules, macromolecules. To bring these big particles into the cell or to get them out of the cell, you need ATP. So this is active transport, vesicular transport, transport of large particles. There are multiple types of vesicular transport. Endocytosis, which means endo means within, cytosis means cell, endocytosis means transport into the cell. And there are 3 types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. We'll talk about 2, phagocytosis and pinocytosis. To get things out of the cell, it's called exo. Exo means out, cytosis means cell, exocytosis. Within the cell, it's called transcytosis. How does that bag go across the cell? That's called transcytosis, transporting within the cell. Endocytosis. Endocytosis involves the formation of vesicles that are coated with protein. It is very selective. It involves receptors. Substances being pulled in must be able to bind to its unique receptor. One of the typical examples of endocytosis is how to get glucose into a cell. Glucose has to bind to insulin, and insulin will get the glucose into the cell. Without that receptor, glucose cannot come into the cell and will stay in the plasma. So if you have no insulin or a low amount of insulin, that means you're diabetic. That means your blood sugar in your blood plasma, which is extracellular fluid, will be very high. The normal blood sugar is between 70 and 110. And if you lack insulin or have a low amount of insulin in your cell, and you can't get sugar inside your cell, your blood glucose would be over 110, sometimes 6, 7, 8, 900. So you need insulin to get glucose into the cell, and that's called endocytosis. Endocytosis. Okay? So that's important. What really happens is this particle that you need to get into the cell, it fuses with a bag of hormones or enzymes, and then it comes into the cell. Here's a typical example. Here's something that needs to get into the cell. It's coded. I think there's a thing here. Yeah. It's coded to ingest. It ingests the protein coat, and it goes into the cell. And is utilized, and then is thrown out of the cell. Okay? So it's brought in by a vesicle, a bag of enzymes. And it goes into the cell to be used. And once it's used, it now leaves the cell. So that's important. Another type of vesicular transport, and a very important one, is known as phagocytosis. Okay? This type of endocytosis is basically known as cell eating. Phagocytosis is what your macrophages and white blood cells use to kill bacteria. They eat bacteria. So what they do is they form a bag, they grab the bacteria, and they bring it into the cell so it can be destroyed by the enzymes inside the cell. So phagocytosis is used by macrophages and certain white blood cells. Here is your bacteria. Here is phagocytosis. This bag, it's called the phagosome, and it engulfs the bacteria and digests it. Okay? So now we have another type of endocytosis called pinocytosis. Pinocytosis means to drink water. Pinocytosis is the bringing in of fluid into the cell, water into the cell. The plasma membrane infolds, bringing extracellular fluid and dissolved solutes into the cell, into what's called an endosome. It's like these cells take a piece of the outside environment and bring it into the cell. It's the main way in which the cell has nutrient absorption. And when you say the word nutrient absorption, it always occurs in the small intestine. This is how the small intestine gets nutrients into the blood, by pinocytosis. Here it is. They indent and they bring in fluid from the outside world and also important solutes. Now, these solutes can be amino acids or proteins or any such things like that. Okay? Receptor-mediated endocytosis is extremely rare and basically only occurs with multiple diseases. You really don't have to know anything about receptor-mediated endocytosis. That's not important. I'm not going to ask you any questions on RME, but I am going to ask you questions on exocytosis. How does the cell get rid of things? Well, it gets rid of things through exocytosis. It forms a secretory vesicle and throws it out. literally throws it out. It gets rid of hormones, it gets rid of mucus, it gets rid of waste, and it gets rid of neurotransmitters. Okay? That's exocytosis, getting rid of things. That's basically all you have to know. You don't have to know snare proteins. That's not important. But this is what's happening. It's like a it's like a pimple burst. Here it is. They're getting rid of it. This is the endosome, which is getting rid of things out of the cell. A secretory vesicle. Resting membrane potential. Resting membrane potential is the electrical potential energy produced by the charged particles across the plasma membrane on all cells. If a cell has a charge, it is said to be polarized. The difference in electrical charge between 2 points is known as voltage. The one thing I want you to know is that the majority of membrane voltage is between -5 to -100 millivolts. That means that the inside of the cell is negatively charged to the outside of the cell. It is potassium that has the major role in the resting membrane potential. The electrochemical gradient of potassium sets the resting membrane potential. Most cells, because of potassium, have a resting membrane potential of -90 millivolts. I'm going to ask you, what is the key player in the resting membrane potential? The answer is potassium. I'm going to ask you, what is the resting membrane potential of most cells? The answer is -90 millivolts. Sodium has an effect, but a minor one. When sodium enters the cell, because it's positively charged, it will bring the resting membrane potential up to -70. So it adds +20. When you add +20 to -90, you get -70. So it brings the resting membrane potential up. Chloride has little influence and should not be worried about. Just remember that sodium, because it's positively charged, will bring the resting membrane potential to -70, which is up, closer to zero. Okay? Closer to zero. I'm not going to ask you any questions on CAMs or plasma membrane receptors. I'm going to go right through it. You don't have to know this. Not important. And I think we're done. Let me stop the audio, the recording here.